Hi class, in this recording we are going to focus on PhysioEx exercise number 6, activity number 1. And if I flip and go to your lab objective worksheet for this semester, or for this week, we can see that for activity number 1, let me adjust my little writing stylus there we go. For activity number 1, we're going to be skipping question number 1. So as we go through our explanation, I'll make sure to skip that question. So let's look at activity number 1. During activity number 1, we were focusing on the refractory period of cardiac muscles. Now, in skeletal muscle tissues, we can have wave sublimation occur. So as we increase the frequency of stimulation of a skeletal muscle cell, we can increase the tension, and the tension waves will begin to overlap with each other, and eventually we can reach tetanus or tetany. However, in skeletal muscle cells, if we have tetany, we, generally speaking, aren't going to die. I mean, if your leg's cramping up, it's not going to kill you. It's uncomfortable, but it's not lethal. And we can get away with that in skeletal muscle cells because skeletal muscle cells have a short absolute refractory period. However, as we look at cardiomyocytes have a big plateau phase to the cap graft. And that means that cardiomyocytes have a very long absolute refractory period. And the relative refractory period of a cardiomyocyte is also relatively long. I know I said relative twice there. So when we think of a cardiomyocyte, really to think that the big take-home picture is that skeletal muscle cells can initiate action potentials at very high frequencies. Cardiomyocytes have theoretical maximums that are much longer for initiating, or that are much longer in between adjacent action potentials. This means that there are mandatory rest periods in between action potentials for cardiomyocytes, primarily because of that plateau phase on the cap graph. If you haven't watched the video of the cap graph yet, I encourage you to stop now and go back to the other video I just made on cap graphs. I'm assuming at this point that you've already watched it. So as we look at our setup for this activity, in your experiment, you're given this experimental setup where we have a heart and some electrodes, and the heart's being stimulated by the electrodes, and then you are going to also be recording the ECG, the electrocardiogram of the heart, and then collecting some data. And as during the experimental procedure, this experiment for PhysioEx exercise number 6 looks like this, where you can take electrodes and go, hey, and give multiple stimuli or single stimuli and change the action potentials so that the EKG or the ECG looks different. And then while working through this activity, you end up collecting some data that, let me pull it up for us right now. And as we collect that data, desktop, PhysioEx activity 6, here we go. You end up with these two graphs when you're done with your experiment. So we have a graph that looks like this by giving a single stimulus to initiate extrasystole. Extrasystole is right here. This is the extrasystole. So we have an extra contraction of the ventricle right there. And then for multiple, when we have the multiple stimuli, stimulating the heart in the experiment with that 20 stimulations per second, we can also initiate extrasystole as well. But we also are going to have phases where there is no contraction of the heart. Let's go back to our post-lab questions. So we are skipping, according to our assignments, we're just going to skip the frog heart, because really I don't care about frog hearts. This is human A&P, not comparative A&P. But when we look at that extrasystole corresponds to an extra contraction of the ventricle. And in this simulation, we're using a frog heart, so theoretically there's one ventricle as opposed to a human heart with 2 ventricles, so that we have an extra contraction of the ventricle. And we induce extrasystole by giving the heart a stimulation, by stimulating the ventricle while the ventricle is relaxing. Let's go back to the cap graph. We need to give a strong electrical stimulus during that repolarization phase. And during that repolarization phase, we have the relative refractory period where it's possible to initiate a new action potential. We just need to use an electrode on the heart because we need a really strong stimulus for it to occur. Let's look on to question number 3. As we look at question number 3, it asks, why is it important that wave summation and tetanus do not occur in cardiac muscle or cardiomyocytes? It doesn't take a rocket scientist to answer this one. If your heart cramps, if we have tetany of the heart so that the heart muscles just seize up and stop moving, then your heart stops pumping blood and you die. So it's important that we don't have wave summation and tetanus in cardiomyocytes because it kills you. We have a strong selective pressure against doing that, because if that was a problem for an individual, that individual probably is not going to be reproducing and passing that trait on to other generations of human beings. So that's all we have for PhysioEx exercise number 6, activity number 1. If you have any questions or comments, please feel free to post them in the class discussion board. And as always, happy studies.